from the University of Waterloo, which is about an hour from Toronto. So we are very close to a major center that's interested in nanomedicine and bio-nano types of studies. First of all, I would like to thank Dr. Lee and ISNM for the invitation to speak here and present some research and overview of some nanoparticle-related material. ISNM is the American Society for Nanomedicine, and it's one of the sister organizations in the world that works together with every other nanomedicine societies. And I just wanted to bring your attention that there are groups in North America as well that are interested in pursuing and hopefully translating nanomedicine to the clinic. In Canada, as I said, we are located in Toronto, but around Montreal, Calgary, and in Alberta and in Vancouver, there are nanotechnology institutes that are interested in nanomedicine development, nanomaterial development, as well as clinical nanomedicine. As far as my research group, we are interested in developing nanomedicines for non-invasive drug administration, and we use different soft materials to build nanoparticles, nanodelivery systems. These can include lipid vesicles, Gemini nanoparticles that are based on cationic surfactants. We develop permeation enhancer libraries so that we can enhance penetration of the nanoparticles or drugs across barriers like the skin, the cornea, gastrointestinal system, nasal mucosa, and other mucosal surfaces, for example. We also use lipid conjugation to enhance hydrophobicity and crossing hydrophobic barriers, and carbon nanotubes as well for attachment of nucleic acids and transporting nucleic acid and genetic materials. So this is sort of the overview of my group. And as you can see, these have many applications for non-invasive administration routes, including dermal, transdermal, intranasal, ocular, buccal, and oral, intravaginal, and pulmonary administration as well. One of the areas that we have made a lot of progress is developing biphasic vesicles as dermal and intravaginal delivery system. These biphasic vesicles are phospholipid-based vesicles, and they entrap proteins to transport through the skin and mucosa. And this type of system was developed as a cream for topical administration or for intravaginal administration for the treatment of human papillomavirus infections, for genital warts, and the prevention of cervical cancer. This formulation is being investigated clinically for two indications. One is anogenital warts, and the other is a low-grade cervical lesions for CIN1 lesions for HPV patients. And this is the one indication that was approved for phase 3 studies recently. Today I want to give you a little bit of an overview about innate immunity to nanoparticles, and this is really going to be a research overview where I wanted to give you kind of an introduction to all the types of things that can happen to nanoparticles when they enter the body, whether it's going to be injected intravenously or inhaled or applied topically, or maybe subcutaneous or intramuscular injection when the particles encounter interstitial fluid. So there are many ways of administering these particles, and the body will react to them. The nanoparticle properties, of course, will be very important. So we'll talk a little bit about what can happen, what kind of processes can go on, and distinguish between two major goals that we want to achieve with these nanoparticles. One is if we want to use them for treatment purposes, then we actually want to have immune neutral particles. We don't want the body to react or minimize this reaction, so there would not be any immunogenicity. What kind of strategies are there for that? Nanoparticle designed for vaccine purposes, where the ultimate goal is specific stimulation of the immune system and achieving protective immunity. So what can we do to aid the recognition and the presentation of the antigens, and some overall perspectives. My perspective on this, of course, will be from a drug delivery scientist point of view, so not quite just immunology or just clinical, but the design of the systems as well. So there are different kind of modalities, treatment modalities, delivery systems, and vaccines and diagnostic devices that are based on nanoparticles. We look at a little bit of timeline, nanomedicine types, and their significance. Now, let me just start with this, because when we talk about nanoparticles, there has to be some kind of context, and it all starts with how the material changes when the particle size decreases. When the particle size decreases, then all of a sudden there will be a lot of surface area proportionately to all the molecules that particle contains. So by just decreasing nanoparticle size from 100 nanometer to 10 nanometer, we are changing the percent of surface atoms from 1.5% to 15%. And another decrease, a tenfold decrease, would result in nanoparticles that where 90% or more of the atoms are now on the surface of that particle. So this is a huge increase in surface area as the particle diameter decreases. This will, of course, have consequences. We have to deal with those surface effects, what's happening on that surface, and this actually invites all those possibilities that a nanoparticle will experience when they enter the body by different routes of administration. Nanoparticle types and the nano entities, engineered nanoparticles or biologics can also be categorized into the nanoparticles that we have to deal with in terms of understanding their interaction with the body. So as we all have seen a lot of reviews and a lot of summaries for these, there will be different particles made from different nanomaterials. They have all different properties, and not just simple carriers, but now we have a lot of very sophisticated carriers where there would be surface groups attached to it for various reasons, whether it's targeting or for other types of decoration, shielding away from certain targets, hiding them. So there would be a lot of variety of different particles. Then we have all the proteins that are administered into the body, those that are down 5-10 nanometer size, like an antibody would be about 10 nanometers, and all the varieties of different antibody derivatives. And of course the nucleic acids, which can be the plasmids, oligonucleotides, siRNA, mini-strings, mini-circle, plasmid DNA that's used for gene therapy purposes, and nanoparticles can be made out of them as well. Now, as we move in time, we have come a long way from the 1960s when liposomes were discovered, As you can see from this diagram, which was done probably 5-6 years ago now, so we don't even have some of the really new ones on this particular diagram. But you can see that pegylated liposome was becoming available in the 1990s. There were modifications to that. There are microneedles that have been developed for administering particles through the skin. There were different types of decorations and encapsulation methods, dendrimers that have been developed, the print technique to encapsulate agents, which makes possible making very specific shape particles that we can utilize to, again, mount certain kinds of responses. So when we build nanomedicines, from pharmaceutical point of view, from pharmaceutical development point of view, we always have to look at our materials, nanomaterials, as pharmaceutical excipients. They have to comply with certain toxicological requirements for approval by regulatory agencies. When we use them, then we can create delivery systems from them. So, for example, a phospholipid can turn into a liposome, or we can utilize polyethylene glycol or gold particles, and their combination or association through self-assembly could provide us with various types of nanoparticles. The drug substances, of course, would be small molecules. It can be DNA, it can be proteins. The potential products that we develop from them can be various dosage forms. These dosage forms can be liquids, they can be semi-solids, they can be solid dosage form depending on the route of administration. So they can be used for different routes such as oral or ocular or injectable administration methods. And at this point, these medicines can be considered generic medicines, and when an innovator product comes on the market, it would be an innovative nanomedicine. For example, the first one, Doxil, approved in 1995. So since that time, of course, there are a number of different nanomedicines that have been approved and available for use in the clinic, and we are expecting a lot more and taking advantage of the principles of nanotechnology. The importance and challenges of engineered nanoparticles in nanomedicine, of course, include so many things that we have to take into consideration. So we gain in terms of improved drug delivery, targeting, controlled release. We can design these particles in such a way. We can have multifunctionalities and great flexibility for tailoring specific disease applications. But we also have to deal with potential immunotoxicity reactions, allergy, anaphylaxis, hypersensitivity, or immunosuppression. We also have to understand that the properties are complex. There are unpredictable reactions, and we still lack materials that would be truly safe and efficacious at the same time. Sometimes you get efficacious but not quite safe. Sometimes you get something is quite safe and it doesn't really work all that well. So we have to find just the right combination for clinical translations. Among the approved nano-based products are different, of course. Liposomes are the most frequently occurring in this group, but there are some other types of nanoparticles, nanocrystals, micelles, spheos, nanoemulsions, dendrimers that are also coming in for clinical application. Now, when these types of drugs are developed, then the ICH guidelines will require various immunotoxicity studies for human pharmaceuticals. Most of these are included in standard toxicity studies. However, there is a requirement now for additional immunotoxicity studies based on either the results that are coming out from the standard studies, or because depending on some knowledge about the material, that there would be some anticipated reactions, so then you would have to start testing for that. And some of the reasons why this scrutiny is happening is because of what the particle is going through, and we will look at the reason for that. Now, in about, it was around 2007 or so when the first paper came out about interactions of nanoparticles in plasma, and findings that some kind of proteins from the plasma can adsorb to the surface of nanoparticles, and changes can occur with these nanoparticles. The fate of the nanoparticles can be different. So with this paper and subsequent work, the bio-nano interface all of a sudden started to become very, very complex. A lot of studies in the past 10 years or so revealed just how complex the picture can be. When you look at the particle surface, then you can see that on the surface there will be different binding interactions. There would be size-dependent changes, and depending on particle size, there would be different kind of adherence to different proteins in the plasma. There could be charge interactions, hydrophobic, hydrophilic type of interactions. Also, there may be acceleration of the dissolution of the particle material due to protein binding. There will be different kind of adsorption and desorption phenomena. That means there is a dynamic interaction between the milieu and the protein and the particle surface. And all of these can be concentration dependent, material dependent. So there is a lot of complexity with these materials. The biophysical interactions were easily described initially by looking at physicochemical principles, colloid stability phenomena. So some of these interactions were understood based on physical chemistry. But a lot of the changes that were occurring because of this adsorption of the proteins on the surface was not only the physicochemical changes, but also immunological changes. So what happened was that the immune system recognized these particles now in a different kind of manner. So the innate immunity, which is really the immunity in the body that's always on standby and would recognize any invader entering the body, and it contains proteins and it contains certain cells that are capable of immediately reacting and removing any particle or pathogen that comes in as a foreign particle. So you would have all the granulocytes, dendritic cell macrophages, natural killer cells, and proteins in the plasma, and the complement proteins, even proteins that is very abundant like albumin, lipoproteins, various lipoproteins. And so these cells and proteins would react and try to eliminate anything that's recognized as a foreign particle. The adaptive immunity will be important when we talk about particle designed for vaccine purposes, because in that case you would actually want the innate immunity not to recognize, let it go through the particle, and then there would be either a humoral or cellular immune response mounted to the antigen that we are trying to present for vaccine purposes. Now, It's so important to recognize how nanoparticle properties determine their interaction with the immune system. So here you see kind of a brief summary of what are those major factors like size, charge, hydrophobicity, targeting moieties that could be on the surface of the particle, that all play a role on whether there would be immune stimulation, there would be adjuvanticity of those materials that we administer, how hydrophobic it might be, what is the charge, is there a charge interaction, and how that influences particle behavior, and of course the specificity of the ligands that are on the surface. When these types of particles are administered into the body, they may change biodistribution depending on what's on their surface. When we make a nanoparticle and we have in the laboratory in our little vials, particles, and we characterize them, that's one set of characterization. The other set of characterization is really environmental characterization from the actual environment these particles would be located. So in a very simple way, the fate of the nanoparticles would have to be divided into at least 3 categories. The bare nanoparticle would be transformed into a particle that is surrounded by some kind of proteins that are adsorbed to the surface. In the first case, we want to have particles that are immune neutral, so they would be safe. In the second case, there would be some adsorption of material, or we would add materials to be immunogenic, which would activate the immune system. And in this case, toxicity could occur. And finally, in the case of vaccines, immunogenic particles would have to be prepared in such a way that to activate the immune system to achieve a vaccine. Now, nanoparticles interact with the immune system and activate pattern recognition receptors, and there are a number of them that have been discovered now. The very first group was the atoll-like receptors, but there are a number of other groups, the NOD-like receptors, or the C-type lectin receptor, or RIG-like receptors that are also now described. And these PRRs recognize molecular patterns, either pathogen-associated molecular or damage-associated molecular patterns, or PAMs or DAMs. When these are recognized, it will lead to, of course, an immune reaction, and this is the signaling pathway how the body deals with the recognized particles, and would mount an immune response and either resolve the situation, or there could be a chronic inflammation that could entail if some of the mechanisms, one of the pathways is not working properly. So some of these pathogen-associated molecular patterns include LPS, flagellae, lipoproteins, zymozyme, nucleic acids, and all these would have their own specific signaling pathway, for example, LPS through TLR4 receptors, and through interactions in the signaling pathway through NF-kappa-B, there would be pro-inflammatory cytokines generated and inflammatory response. And in all of these cases, the complexity is there, and of course, depending on what kind of surface properties the particles have, they would lead down a pathway that would lead to inflammatory disease, autoimmunity, or resolution of infection. Now, in a recent paper, Fadil actually coined a new phrase, which he called NAMPs. And this is something that was asked because if pathogens have PAMPs and cells that fall apart and damaged and need to be cleared from the body are DAMs, then there may be some kind of nanoparticle associated molecular patterns, and whether how and can these interact with the immune system in some way that we can understand these interactions a little bit better. The nanoparticles encounter their in vitro and in vivo environments and develop a biomolecular coating, or also called corona. So now the biomolecular corona is receiving a lot of attention to try to understand its complexity, what kind of proteins participate in there, how can we analyze them. There could be opsonization, complement activation, cytokine response, inflammation. So all these things can happen to the nanoparticles. When a corona develops around the nanoparticle, then you can see that more and more proteins can adsorb to its surface. So technically, once this corona develops, that's the kind of nanoparticle the cell will see. So the cell is not seeing the particle you made and it's in the vial, but it sees an entity that's already changed. So what they say is that this particle changed identity already, and the cells see a much more complex entity compared to just what we intended to prepare. Now, a lot of the properties of the nanoparticles will contribute to this development and the type of corona. The nature of the material, many different materials we'll use, size, shape, surface functionalization, surface charge, all contribute to this corona development. So technically, there is no more naked particle in the body. The corona formation is very dynamic. As we said, many of the properties that we already characterize in vitro would contribute to this development of the corona. There is an inner and there is an outer layer of the corona. Sometimes it's described as a hard corona and a soft corona. The hard corona is the tightly bound layer, and the soft corona is the loosely bound layer where there would be more exchange. But both layers can actually undergo changes depending on the concentration of the proteins in the environment and also their affinity for that particular material. So first, the proteins that are present in high concentration would bind. But as time goes on, they can get exchanged to low affinity protein, these lower affinity, but to the high affinity proteins, even though they are in lower concentration. So there is going to be a change over time until the corona stabilizes. So you will see that CNTs, iron oxide, liposome, polymeric nanoparticles, very frequently have albumin, immunoglobulins, complement, fibrinogen, and apolipoproteins bound to them. As I said, dynamic corona can be in the blood. Of course, there is an abundance of proteins there. But also, if you look at all the other administration routes, so in the ocular environment, you would have different sets of proteins. In the bronchial fluid, you will have different sets of protein. So there will be differences in what kind of properties the particles will have. Now, this table here is not really for studying them in detail, but to impress upon you that there is a different composition of corona in different environments. So depending on where you put the nanoparticle, where the administration is. And there is it will be all different for different particles. And so technically, every particle is going to be unique, and characterization has to be carried out for the types of particles that is being developed. A lot of the methods related to understanding the corona will have centrifugation involved, some kind of chromatographic method to isolate the protein, understand what kind of proteins are there, mass spectrometry, 2D gels. These are analytical methods that will have to be used. Now, quite interestingly, the recent publication of the new version of the plasma proteome database revealed now that especially compared to the previous version, which was 9-10 years ago, the number of total proteins in the database increased from about less than 4,000 to over 10,000 proteins. And these are the number of proteins that found in plasma and serum. And now it's being in the database available to be used to understand their properties and being able to compare the findings from research studies to understand that. But it also kind of implies that a nanoparticle can encounter more than 10,000 proteins, and some of these would be able to absorb to the surface, and which one and how much of it and how long. So there is a lot of pharmacokinetic aspects to this as well. Now, I prepared some observations, very interesting observations that came out just to get an idea of what kind of protein absorb mostly on what kind of nanoparticles. So as you can already see from before, albumin and fibrinogen are very frequently absorbing to all kinds of nanoparticles. Cationic lipoplexes and polyplexes show strong albumin binding. Albumin is negatively charged, so that would be natural, but there could be other hydrophobic types of interactions as well. Positively charged particles prefer to absorb proteins with isoelectric points less than 5.5, and negative surface charge enhances above 5.5, such as IgG. Transferrin, haptoglobule, fatuin alpha, kininogen, histidine-rich glycoprotein, clotting pathway factors are attracted to polymeric nanoparticles, for example, or particles with very hydrophobic surfaces. So what you can see from these kinds of observations is that some patterns start to be emerging. I think we are a long way from really saying that we understand everything, but there is some commonalities between different particles. Apolipoproteins are preferentially absorbed onto liposomes and polymeric particles, but not inorganic nanoparticles, for example. If you have dextrans or sugar hydroxyl groups, this promotes binding of the CC complement. Mannose binding protein binds to phosphatidyl inositol type of liposomes. Dextran-coated particles, when dextran is used to actually shield particles, they can be recognized by antibodies as well. And hydrophobic or charged surfaces tend to absorb more proteins and denature them with a greater extent than neutral or hydrophilic surfaces. This table summarizes to some extent of the major observed effects. Higher charge density of the nanoparticle will increase the corona, and higher hydrophobicity will do the same. The higher curvature of the nanoparticles while increases the corona, but there will not cause conformational change, whereas charge and hydrophobicity can do that. Higher protein concentration in the environment also increases the thickness of the corona. This couple of slides that I wanted to show you is switching from IV administration from the blood to the respiratory tract. And in this study, Kumar and co-workers looked at two different nanoparticles, and they wanted to compare if there was any difference in the type of corona that developed in the presence of respiratory tract lining fluid. And between the two particles, they observed that 400 proteins were commonly absorbing to both particles, but there were unique proteins that absorbed to each of them. The polyvinyl acetate type of particles absorbed a lot more of these unique proteins compared to the silicon type of nanoparticles. So what we have seen from these studies is that there will be proteins that preferentially absorb. So there will be pulmonary surfactant associated proteins, serum albumin, apolipoprotein A, that would definitely absorb to the surface. And then there are some unique ones depending on the material. And with regards to innate immunity, they found that there were differences between the two particles, even though they seem to absorb some similar types of proteins on their surface, but in the different proportions. And so this also would mean that there could be different responses once these particles administered into the body. One might end up to be not as recognized and safer compared to the other one. Now, some current issues, these are some of the little bit of the hot issues with nanoparticle design in the last few years. These are related to stealth coating with PEG. We hear a lot about PEG in the conference and in the literature. Hydrophobic surfaces, the significance of cationic charges, particle size, particle shape. There are some good recent reviews about that as well, and opinion papers. With regards to the coating with polyethylene glycol, which is really the gold standard to increase the circulation time of particles in the bloodstream, it also improves physical stability and reduces non-specific protein absorption, but a lot of the issues are also related to increased recognition by the immune system. Anti-PEG antibodies have been detected, recognition by the complement system, and some accumulation in intracellular compartments that will increase toxicity of PEG-coated particles. Now, I have some few cases that I wanted to show to demonstrate some of these issues. And when we have, for example, an idea, a general idea how, for example, PEG or other polymers would assist in increasing circulation time, then we also see some exceptions, and we have to look at them as whether are they serious, how should we understand them, and does that tell us new information that just wasn't known before. So in this study, it was found that when the PEO, polyethylene oxide type of polymer, was coated on the surface of particles, then by just changing the conformation of these polymeric chains on the surface from mushroom to brush configuration or conformation, there would be a complete switch what kind of complements would bind to the particle surface, and there was a switch from the C1q dependent classical pathway to the lactin pathway, which was, if you look at the B and D particles, there would be a big difference in terms of the rise of the C4D complement in these experiments. And this also means that the clearance of these particles would be different, whereas the first particle, the mushroom configuration, would be cleared through the liver, the other one would not. And the rush configuration actually will be cleared through the spleen eventually at a high degree. So there will be differences there. With regards to carbon nanotubes, which we have known for quite some time that they develop quite a thick corona after administration, a lot of chemical functionalizations that have been tried to overcome this effect and to evade epsonization. So you will see all kinds of non-covalent and covalent functionalization that was done to carbon nanotubes. And indeed, some of these functionalization improved solubility, improved recognition avoidance, but also if you look at just the degree of information that we need to get when polyethylene glycol modified carbon nanotubes are looked at for effect of biodistribution and whether we can shield them from recognition, then the density of the packing of these polyethylene glycols, the length of the chain would be very important. And when you look at the ones that are branched and long chain, they would be always better compared to other ones that are not as densely packed or have shorter chains. On the other hand, you have studies recently which also looked at the effect of the pegylation on single-wall nanotubes. And the group by Saketiet and co-workers looked at scarcely pegylated nanotubes versus highly pegylated nanotubes, and they found great differences, but not the way that you would have expected. So the highly pegylated nanotubes would be excreted fairly fast, and the ones that were hardly pegylated would have slow excretion, and they actually accumulate in the river. They were taken up quite rapidly by macrophages, and you can see the organ accumulation of these kind of nanotubes there. So it's quite interesting how the kinetics would change after this, and this was quite unexpected because you probably would have expected the opposite effect. One thing is not clear from the study, that the achievement of the different degrees of pegylation was through non-covalent versus covalent addition of the PEG. Whether this also makes a difference, this wasn't quite explored in the study. Now, talking about hydrophobicity of the particles, in this very elegant study, I think, oh yeah, okay. It was by Moyano and co-workers, and they were quite interested in looking at just this effect of hydrophobicity. So they prepared a series of gold nanoparticles with various hydrophobic groups and log P values, and they wanted to see if these particles would activate the innate immune system, and looked at cytokine expression in splenocytes as a function of the hydrophobicity log P. And they found a linear relationship between gene expression of the cytokines and the hydrophobicity. So basically, the more hydrophobic the particle was, the more recognized it was by the immune system. Now, the surface charge of the corona can be quite important as well. And in this study, a couple of years ago, the group prepared amine modified and carboxylate modified positively and negatively charged particles. And they wanted to see what will be the result of the absorption of just albumin, bovine serum albumin to the surface. And quite interestingly, the albumin bound to both positively and negatively charged particles. But what was interesting, that when the BSA attached to the positively charged particles, then you also could see a change, a conformational change in the BSA, and actually the protein got denatured on the surface of the positively charged particles. And as a result of this, it completely changed the uptake mechanism for the particles. So instead of binding to the cell surface through albumin receptors, it is now being cleared by scavenger receptors by macrophages. So these kind of interactions would tell us that we have to be very aware of the particle surface properties and possibility that the behavior will change because of the denaturation or conformational change of the adsorbed protein to the surface. Carbon nanotubes are known to interact, of course, as I said, with various types of proteins, and they are, of course, surrounded in the plasma with various types of proteins. In one study, 4 different proteins were evaluated for their binding to the single wall nanotube surface. And as you can see from the atomic force microscopy studies, as well as from the dynamic modeling, that fibrinogen, gamma globulin, transferrin, bovine serum albumin will create different patterns on the surface of the carbon nanotube. Fibrinogen is initially looks like it's loosely bound, but then it kind of forms ropes and adheres to the surface very well. Gamma globulin would form a uniform kind of structure on the surface. Transferrin is quite organized, and bovine serum albumin is little spherical particles line up on the surface of the nanotube. What was interesting that in the presence of all of these proteins, once the carbon nanotube was coated up, compared to the bare nanotube, the toxicity went down. So the coating improved the toxicity profile for the carbon nanotubes. So all of them appear to be less toxic compared to the bare nanotubes. What also was obvious in the study that the toxicity improved as a result and as a function of the strength of the binding of the protein to the surface. So the actual order of the toxicity decrease was as the result of how strongly the proteins bound to the surface. And so the BFG fibrinogen was the most strongly bound, so that seemed to benefit the most from improvement in terms of its toxicity. There is another very interesting phenomenon that's related to the corona on the targeting ability of nanoparticles. In this study by Salvati and the group, they actually prepared nanoparticles specifically for targeting, and they have a PEG portion, and at the end of the PEG there is a transferrin for targeting ability for these nanoparticles. So what they were interested in finding out that now if we put these ferritin targeted nanoparticles in the biological environment, the corona will develop on the particles, and what will happen? And what they found out that in their study, that once the corona developed around the particles, the targeting ability of the ferritin was completely gone. So it shielded off the entire very elegant work that went into making these kind of nanoparticles with the PEG and the targeting moiety, and it was all gone. Even though particles were still taken up by non-specific phagocytosis, but the targeting function disappeared. Now, we arrive to the point where we look at nanoparticle design for treatment purposes, where the ultimate goal is immune neutral nanoparticles. So what strategies we can have to avoid recognition? We can use various kind of stealth materials. PEG is still somehow in the game. The new materials are the self-peptides that hopefully will help achieve that. Then red blood cell and cancer cell membranes then can be used to coat particles. And some inorganic nanoparticles calcium phosphate, for example, and other natural polymers may be candidates to have immune neutral particles. This concept of avoiding recognition goes to the Disher group in the University of Pennsylvania, where back in 2008, they recognized that the CD47 protein that's present on almost every cell surface, a cell membrane, can provide a peptide that could act as a minimal peptide passport, because if the particles have this peptide displayed on their surface, it signals to macrophages that I am self, so don't eat me, as opposed to the ones that don't have this signal and they would be scavenged in the system. So in their study, about 5 years later, they actually created nanoparticles with this very specific self-peptide that came out of the CD47, which binds to the SIRP-alpha on the surface of the macrophage. And this passport allows the particle not to be recognized. Because of this, the circulation time of these particles in the body increases compared to the ones that don't have this self-peptide. So these minimal self-peptides will hopefully help us, that can be grafted on other particles as well, to improve their residence time. Now, for vaccine purposes, the ultimate goal, however, is stimulation, recognition in the appropriate manner, and in such a way that we can achieve protective immunity. Vaccine design always looks at preventative and therapeutic aspects, and there are a few strategies that can be used to aid recognition and presentation. Some of these are microbial mimicry, immunostimulatory biomaterials, co-delivery of antigens and adjuvants, and controlled release. The pathogen sensing by the immune system is, I'm a little bit repeating the earlier information, but the immature dendritic cells will constantly sample the environment, and depending on whether there is immunogenic cell death or tolerogenic cell death, there will be recognition of these materials by these cells, and the dendritic cells would help mount either a humoral response or a cellular response to these materials that induced some of the PRRs on the surface of the dendritic cells. In these cases, there is always a factor as to what kind of form the antigen would be in, what kind of nanoparticles, and how they are packaged. There are, of course, a lot of challenges. This table here actually summarizes for the 4 different categories of vaccine strategies and how we want to build immune protection or eliminate some kind of recognitions for various disease settings. So there are differences when you are dealing with infectious diseases that are related to bacterial viral infections. And the challenges here are rapid mutation rates, or when you don't even know where the neutralizing sites are, how to control certain immune responses. So a lot of the immunological challenges will have to go back to nanoparticles design and how we achieve these particular functions. So I'm moving on to show you a little bit of these strategies. And one group of these strategies is microbial mimicry to design vaccine nanoparticles. So for example, immune complexes on pathogens can be mimicked by repeated presentation of these motifs. And some mice cells or polyplaxes were developed based on this kind of idea. Viruses can be mimicked by the proper presentation, surface display of danger signals, and we have a number of nanoparticle designs that utilize this type of idea. The co-incorporation of antigen and danger signals that based on bacteria, fungi, and parasites would lead to other kind of nano and microparticles that would be able to entrap and at the same time display some surface danger signals on their surface. The particles that are developed for vaccines will have to be carefully evaluated for particle size. Indeed, the design of the vaccine particles would have to be tailored for the right particle size. The reason for that is that after administration of vaccines, which very often happens subcutaneously or intramuscularly, in the interstitial fluid, the particle size will determine whether the particle will get stuck in the extracellular matrix or would be able to move on with the lymphatic flow towards the lymph nodes. So very large particles would, about 500 nanometer or so, they could easily get stuck in the extracellular matrix. When you have intermediate sized particles and small particles, they will be starting to move towards the lymph node, and based on their size within the lymph node, they can be either present in the macrophages, or they would be, the very small ones would go to the B cell zone, but that rarely happens because those small ones get diluted out way earlier on. Or some of them would be in the T cell zone. So the particle interaction in the lymph node would be very much size dependent. But there is also a question about size, whether the size of the actual particle is important or what's inside of it. So if we entrap smaller particles in a larger entity and we use them as an encapsulated entity, then whether those would be considered and at what point in time they would change over to actually small particles from a larger particle. So that's a factor as well. So overall, the delivery methods that can benefit or we can contribute to improving vaccine delivery would be size manipulation, providing a depot effect. Definitely in nanoemulsions or liposomes we could achieve that. Repetitive antigen display and cross-presentation. So these are some of the strategies that nanoparticle design can take into consideration. Now, in my lab, we have developed some vaccine particles for CpG oligonucleotides to actually vaccinate neonatal chickens. And in these cases, the CpG is internalized by the cell to provide immune function, recognized by TLR receptors, and the signaling cascade would lead to Th1 type of immune response. The in ovo vaccination model would allow us to vaccinate very early on, and in this model we can test out based on the protection of chicks against E. coli infection in ovo because the chicks are very sensitive to infection within the first week of their life. Therefore, immunization early on would prevent the loss of chickens in this way. The problems here, and this is what kind of demonstrates the type of strategy. The problem is that the CpG alone has very short-lived effect, so it doesn't have a long-term protective immunity. it can be degraded by nucleases, it has unspecific bindings, and the PRRs for CpG are intracellular, not on the cell surface. Therefore, we need some kind of delivery system that could take larger concentrations inside the cell to achieve longer-term effect. So we have developed carbon nanotubes as particles for immunization. In our case, we have taken carbon nanotubes, functionalized them with cationic surfactants. These are called Gemini surfactants. They are twin surfactant materials, and they wrap around the nanotube. They cationicize on the surface, and we can bind nucleic acids to the surface to be able to carry them. So the particle itself is going to be closer to neutral. After characterization, we use that for vaccination. The second type of particle was a phospholipid particle with the Gemini surfactants, where we can also encapsulate nucleic acid and prepare these types of nanoparticles that now can encapsulate CpGs. So by encapsulation and surface display, we were interested in the effect. I skipped the actual protocol. If somebody wants to know that, we can discuss. But basically what we have found that both nanotube and lipid surfactant nanoparticle system increase the survivability of the infected chicks after administration of these nanoparticles in ovo. Clinical scores were much better compared to the non-encapsulated particles. So definitely even clinical benefits were obvious, and the bacterial cultures also showed that the formulated CpG groups had a number of birds that had a lot lower bacterial infection compared to the control groups. The other types of delivery system that we have developed was biphasic vesicles as a protein delivery system. And in this case, we wanted to use these for vaccinating against Yersinia pestis for an intranasal immunization protocol. The reason for the preparation of this kind of vaccine is because after administration through an intranasal route, mucosal immunity can be elicited. So by changing the administration route from originally intramuscular immunization that protected individuals against the bubonic plague, now after intranasal administration, the pulmonary form of the plague that can be used for military purposes, these soldiers didn't have to go through the excruciating intramuscular injection, but through inhalation, they were protected against both forms of the exposure. So by encapsulating in biphasic vesicles, lipid vesicles, the survival of the animals was greater than any of the controls. So as a conclusion, many types of immune responses. The corona is very complex and unique for each particle, and the purpose can be immune neutral or immunogenic based on the clinical use. Wanted to give you collaborators, and I want to thank all of them for participating in our research. Thank you. Thank you.
Information related to the nanomedicine, especially how the nanomaterial is supposed to the bloodstream are effective in terms of PCR-free diagnostics.
Thanks for the introduction. I'm Dal Hee Min, and today I'd like to talk about the bio applications of nanographene and graphene oxide, especially for the development of biosensors to be applied in the biomedicine area. So my lab is focused on the development of biofunctional nanomaterials and application of those materials for the biosensors and drug delivery systems, especially gene therapy and protein therapy. So for the nanomaterial side, we are very interested in proposing new structures of porous nanomaterials. For example, porous silicon nanoparticles and porous gold nanoparticles. Porous silicon nanoparticles are very attractive material because we can encapsulate RNAs or DNAs inside these pores so that we can protect those fragile nucleic acids from the nuclease attack, and we can increase the half-life of the encapsulated biomolecules, so that we can apply these materials for the delivery, intracellular delivery and of DNA or RNA and protein-based drugs. And in case of porous gold nanoparticles, we have very large surface area of these gold nanoparticles, and we can also encapsulate the fragile materials inside. And in addition to those protective effects, we can provide a very efficient photo thermal effect based on these gold materials. So those are the materials for the development of drug delivery systems. And for the biosensor development, we are actively use these graphene oxide, graphene-based materials. So today I will mostly talk about graphene oxide-based biosensor development. So graphene oxide is always compared with the graphene itself. Graphene has this sp2 carbon network, so it's kind of like interstitial. The benzene rings are like in the 2D arrangement. It's a two-dimensional, one atom big materials, and this graphene has very good electric properties. So electrons in this material can move very freely. So it's conductive, but mechanically very strong. But compared to this graphene itself, graphene oxide has many oxygen-containing functional groups. So it's not very conductive. So electrons are localized here and there. But for the bio applications of these graphene-based materials, I prefer to use graphene oxide in place of graphene because graphene is not soluble in the aqueous solvent. But graphene oxide is freely suspendable in aqueous solvent and various buffers. So in this graphene oxide, we still have hydrophobic surfaces here and there. And at the same time, we have hydrophilic edges and hydrophilic these epoxide groups on the surface. So we can consider this material as a very big amphiphilic molecule. So it has hydrophobic surface and hydrophilic edges. And advantage of using graphene oxide for biosensor application is mainly because of these two factors. One is it has strong binding with nuclear bases through pi-pi stacking interaction and hydrogen bonding. So the exposed nucleic acid, ATGC, those nuclear bases can fairly strongly interact with this surface. But if those nuclear bases are hidden inside of the duplex, they cannot interact with this surface. And the other thing is the graphene oxide can provide very efficient fluorescence quenching efficacy. So if the hydrophobic small molecule-based fluorophores are present nearby this surface, those fluorescence can be very effectively quenched by graphene oxide. So those two properties are very unique and very useful properties of graphene oxide for the biosensor development. So for this, the bio applications of graphene oxide, we prepare the graphene oxide from graphite flake. So we chemically oxidize, and if needed, we further reduce this graphene oxide so that we can recover partial properties of graphene itself. But most of the times we just use graphene oxide form. But of course, the size of graphene oxide and the degree of oxidation, those properties are very important for proper function. So since about 2008 and 2009, many researchers got interested in the bio-applications of graphene derivatives, especially for biosensor and enrichment of the small biomolecules and detection of biomolecules, and also enzyme activity assay. So this is I'd like to introduce one review article which I wrote with Professor Hee Min. Actually, Korea is very strong in this this research field, the graphene, physics of graphene, and also bio-applications of graphene and graphene oxide in this field. So if you want to know more about the biomedical applications of graphene and also graphene oxide, then you'd better read this Council of Chemical Research review article. It's a very nice introduction in this field. If you are more interested in biosensor applications of graphene oxide rather than graphene itself, then I recommend you read this review article, which is published very recently. And this article, we summarize the biosensor applications of graphene oxide according to different detection methods. So the FRED, LDI, SARS, electrochemistry, these are four most major categories of detection of biosensors based on graphene oxide. So FRED is fluorescence resonance energy transfer. So as I said earlier, the graphene oxide can quench the fluorescence very effectively, but those quenching is reversible. So if the single-stranded nucleic acids, which is labeled with fluorophore, are mixed with the graphene oxide solution, then these single-stranded nucleic acids will be absorbed on the surface of graphene oxide. So those the fluorescence can be quenched. But if you add complementary sequences of this nucleic acid probe, then you can dissolve this absorbed single-stranded DNA from the graphene oxide surface by forming this duplex, so that you can get recover the fluorescence signal. So this is main principle of FRED-based biosensor in this field. And in case of LDI, it's a laser desorption ionization mass spectrometry. It's one of the mass spectrometry methods. So the graphene oxide and graphene is famous for very effective photothermal conversion effect. So if we shine the light on the graphene oxide with proper wavelength, then this graphene oxide can be heated so that if the small molecules are observed on the surface of this graphene oxide, then the heat and the thermal energy can be transferred to the small molecules so that these small molecules can be ionized and dissolved from this surface, so that we can get the mass spectrum signals here. So this is also this method, LDI mass spectrometry method, is also very useful for the some kind of label-free detection of biomarkers. And in addition to these two categories, the SARS surface inestro Raman spectroscopy and electrochemistry, these two fields are also very actively studied area. But my group is mostly focused on these two categories for the biosensor development. So if you read this review articles, you can find more detailed explanation about the detection techniques of these four different types, and also the purpose of using these detection methods. So if you are somehow interested in using graphene oxide for your biosensor development, then you better first choose which type of detection method you want to use. And you can develop and you can prepare the graphene oxide, proper graphene oxide, which is appropriate for your own applications. So let me go further about FRED-based biosensor development. So graphene oxide, as you can see here, dye solution itself has very strong fluorescence signal. But once you add graphene oxide to this solution, you can see that most of the fluorescence can be quenched. It's more than 95% of quenching here. And if you use somehow, if you have somehow well dispersed reduced graphene oxide solution, then you can see further more effective fluorescence quenching like this. So you can use this quenching capability of graphene oxide and this recovery of the fluorescence for the sensor development. So my group is developing graphene-based biosensors for mainly two different categories: enzyme activity assays and DNA and RNA detection. So for the enzyme activity assays, we demonstrated that graphene-based materials can be very usefully applied for the quantitative activity assay of various enzymes. So, for example, phospholipase, exonuclease, endonuclease, methyltransferase, helicase, and kinase assay. So we can apply all different sources, search of graphene oxides for these enzyme activity assays. And today I will talk about a helicase assay and antiviral drug screening part among these. And for the DNA and RNA detection, I will introduce about development of quantitative real-time microRNA sensor, which can be applied in living cells. So first, helicase activity assay system development and also screening of helicase inhibitors to treat hepatitis C. Hepatitis C is very serious disease, and if you get infected by hepatitis C virus, it develops chronic disease more than 80%, and also there's no vaccine, so we cannot prevent this hepatitis C. So current treatment includes interferon or combination therapy of interferon and ribavirin, but this current hepatitis C treatment has so many side effects, so we need to develop new therapeutic modalities. If you see the life cycle of hepatitis C virus, you can see any step here can be a target for the drug development, but we are mostly focused on this part, the gene replication part. So if we can block this step by inhibiting essential enzymatic activities which are required for the RNA or DNA replication. For example, polymerase, protease, helicase, those enzymes are three essential enzymes which are needed for the hepatitis C virus replication. So among these three enzymes, these polymerase and proteases are very actively studied in big pharmaceutical companies as a drug targets. But in case of a helicase, no single helicase inhibitor is under development for hepatitis C treatment. So it's very not usual. So I wonder why it is. So I think there's no good helicase activity assay method which is compatible with the high throughput screening. So for traditional way of helicase assay, we just use gel electrophoresis. We can so that we can resolve the restricted DNA, which is substrate of helicase, to the single-stranded DNA, which is product of the helicase. But it's very laborious. It takes a long time, so it's not comparable with high throughput assay. So we need a new assay platform for helicase activity assay. So we thought that graphene oxide would be very, very useful for this purpose. Because graphene oxide can distinguish double-stranded DNA from single-stranded DNA. So if we mix double-stranded DNA, which is labeled with fluorophore, with the graphene oxide, then there's no changes in fluorescence intensity. But once helicase is added and this double-stranded DNAs got unwound by helicase activity, then they form single-stranded DNA. So these single-stranded DNAs can be observed onto the surface of graphene oxide, so that the fluorescence of these fluorophores can be effectively quenched. So by following how fast the fluorescence can be quenched, we can estimate the helicase activity. So this is fluorescence-based helicase activity assay system. So it's quantitative enough. So we show that it's, the fluorescence quenching is dependent on the helicase concentration and also ATP concentration, because helicase activity requires ATP as a cofactor. So, and we demonstrate you that small molecule inhibitors of helicases can be applied to get a very nicely fitted IC50 curve. So we show that this helicase assay system can be used for quantitatively address the kinetic parameters. And also we show that this method is comparable with the high throughput screening. So in the 96 rep plate, we applied the different mixtures of this helicase assay solutions. And after incubation, we get the fluorescence imaging of the whole plate. So that we know that in the high fluorescence, the well shows that very low the helicase activity. So after we develop this helicase activity system, assay system, we further applied this system for the high throughput screening of the real drug target. So we chose two different helicases, SARS coronavirus helicase and hepatitis C virus helicase. Unfortunately, these two helicases recognized two different types of substrate, so that we could perform multiplexed drug screening. So we prepared two different substrates for each helicase, which is labeled with different types of fluorophores. So this multiplexed helicase assay system contains two different helicases and two different substrates and graphene oxide. So if we apply to this mixture, if we apply two different helicases altogether, then we can get all the fluorescence quenching image. But if we apply only one helicase, then the corresponding substrate, only the corresponding substrate can be unwound. So only these fluorophores can be quenched, so that we can get only the fluorescence signal only from this red fluorophore. So this assay is orthogonal each other for the specific helicase. So for the multiplexed helicase inhibitor screening system, we use the 96 well plate, and each well contains two helicase substrate and two helicase enzymes and graphene oxide solution. And in addition to this mixture, we added small molecule library compound from the small molecule libraries. So this, you can see the raw data. This one rectangle shows one 96 well plate, and you can see its positive control are all lighted up like this. And you can see the some spot shows very bright fluorescence, which tells you that this well has a small molecule which can be used as inhibitors of specific target corresponding helicase. Because the helicase activity is inhibited, so the fluorescence could not be quenched by graphene oxide in these wells. So after the screening, we selected 5 inhibitors which can inhibit each helicase. And among those, we validated that 2 of the 5 helicase, the HCV helicase inhibitors were active in the cell-based assays also. So this series of studies shows you that the newly developed bioassay, the quantitative enzyme activity assay systems can be directly applied to the drug screening development. And second part is about development of real-time microRNA sensor, which can be used in living cells without fixing live cells, which is named Pango. So microRNA. Why we chose microRNA? MicroRNAs are small RNAs which are present in the cells, and these microRNAs are very important regulators of gene expression. So some microRNA sequences are abundantly present in certain cells at a certain specific status. So some microRNA sequences are very unique, can be considered as unique biomarkers for specific diseases. So current conventional microRNA detection methods include cloning, microRNA in situ hybridization, something like that. But all these methods are not compatible with live cell imaging. So our strategy is based on the probe nucleic acid interaction with the nano-sized graphene oxide, so that this complex, the fluorescence quenching complex, can be applied to the cell media or just to the tissue. Then this nano-sized complex can readily go into the cells across the cell membrane. And then once they meet the complementary sequences of microRNA, then these probe nucleic acids can be dissolved from graphene oxide so that they can get recovered the fluorescence signal inside the cells. So you can very easily apply this method for live cells. So we can get a very nice fluorescence image of the fluorescent cells, and the fluorescence signal subcellular localization can be also used for providing very useful information about the microRNA, like location of microRNAs. So for this application, we prepared the nano-sized graphene oxide. And first, we showed that multiplex microRNA sensing can be possible in the buffer solutions like this. So we mixed the 3 different probe nucleic acids, which are labeled with different types of fluorophores, and mixed all together with graphene oxide. Then we added 1 microRNA at one time, or 2 or 3 microRNAs at the same time. Then we could get only corresponding microRNA signals at each excitation wavelength. And this data shows it's a sequence-specific signal, and multiplex sensing is possible. And the detection limit was revealed as 1 or 2 picomolar concentration, so it's very, very sensitive without any amplification such as PCR. So inside cells, you can see that in the site, this is the signal comes from nucleic acid probe corresponding to the microRNA 21, which is the most famous microRNAs involved in the cancer cells. So you can see the green fluorescence are shown up in the cytoplasm. You can see that some spots are brighter than the background, but overall, all the cytoplasmic area shows green fluorescence corresponding to microRNA 21. So we can assume that the location of the microRNA 21 is fairly spread out throughout whole cytoplasm. And also it's quantitative signal. So as you increase the induction time of specific microRNAs, you can see the increased the fluorescence signal corresponding to that microRNA. So it has a linear relationship with northern blot data. So this Pango system is also comparable with like previously shown the helicase assay system. It's the fluorescence-based assay system, but it can be applied for the live cell imaging, so it can provide the heteroscopy screening for microRNA-associated drug candidate identification, and also high content screening can be also feasible based on this Pango system. And also diagnostic purpose. For the diagnostic purpose, this Pango system can be very usefully used in place of ELISA platform or POCT because it's very simple to prepare and the signal is sensitive enough. It's more sensitive than most POCT kits, but the system is much simpler, so it has very bright future as a molecular diagnostic purpose of, especially, for example, like the companion diagnostics. For those purposes, this Pango system can be applied to many different disease diagnostics. So today I talked about helicase activity assay system based on graphene oxide and quantitative microRNA sensing in living cells. And actually, I'm giving another talk the day after tomorrow at first ISNM Congress at Korea University. There, I will introduce about this simultaneous sensing and silencing of viral gene and the simultaneous sensing of oncogenic microRNA and subsequent photodynamic therapy. So all related to diagnostic application of graphene oxide-based systems. So if you have a chance, I will see you there at the Hana Square, Korea University, the day after tomorrow. And I'd like to thank all the group members and thank you for your kind attention.
So, giving us very good